Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. Today we will start new video. If you like it, please like, subscribe and comment to receive more videos in the future. Let's start our video today. Here we go.
بسم الله الرحمن الرحيم والحمد لله والصلاة والسلام على رسول الله رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. السلام عليكم ورحمة الله تعالى وبركاته. In this lecture we will talk about gluconeogenesis. This lecture is presented by Elham Omer, MSc Medical Biochemistry, Khartoum University. Let's see the objectives of this lecture first. By the end of this lecture, all the students should be able to define gluconeogenesis, recognize the precursor of gluconeogenesis, identify the major gluconeogenic tissues, explain the importance of gluconeogenesis, describe the conversion of each precursor of gluconeogenesis into glucose, and discuss the regulation of gluconeogenesis. Let's explain the term gluconeogenesis. Gluco means glucose, new, جديد, and genesis means synthesis or formation. So it is the pathway concerned with the synthesis of glucose from new sources. It defined as synthesis of glucose from non-carbohydrate substrate or precursor. الprecursor اللي هي المواد الأولية اللي بنصنع منها. It is the de novo synthesis of glucose occur during fasting state. التصنيع الداخلي للجلوكوز بنسميه de novo synthesis. Glucose either comes from the diet or from glycogen by glycogenolysis in the early fasting or by gluconeogenesis in prolonged fasting. Precursors of gluconeogenesis include lactate, which comes from anaerobic glycolysis, glycerol, which comes from catabolism of triethyl glycerol, amino acids, actually the glucogenic amino acid, and propionate, which comes from catabolism of odd number fatty acid. Major gluconeogenic tissues include liver and kidneys. It primarily occurs in the liver during fasting state. Also, it can occur in the tubular cell of the kidneys, which contribute only 10%. Importance of gluconeogenesis. Some tissues such as the brain and erythrocyte are completely dependent on constant supply of glucose, and those known as glucose dependent tissue, because brain need continual supply of fresh glucose, and RBCs lack a mitochondria. When the dietary carbohydrates are not sufficient, the blood glucose level is maintained by glycogenolysis in the liver and also through the synthesis of new glucose, gluconeogenesis. The pathway of gluconeogenesis is basically the reverse of glycolysis. Glycolysis starts with glucose and end with pyruvate, and gluconeogenesis starts with pyruvate and end with glucose. It is also an endergonic process that consumes ATP. The aim objective of glycolysis is the production of ATP, but this pathway is consume ATP. Pyruvate, which comes from glycolysis, is enter the mitochondria, and instead of converting into acetyl-CoA and Krebs cycle, it converted into oxaloacetate by enzyme known as pyruvate carboxylase. This consumes ATP. Oxaloacetate is converted into phosphoenol pyruvate by phosphoenol pyruvate carboxykinase, also consume ATP. And the phosphoenol pyruvate is intermediate of glycolysis. Go in the reverse reaction of glycolysis until we reach glucose. It first converted into 3-phosphoglycerate and then to 2-phosphoglycerate. The 3-phosphoglycerate is converted into 1,3-bisphosphoglycerate by phosphoglycerate kinase. This consume ATP. 1,3-bisphosphoglycerate is converted into fructose 1,6-bisphosphate. Consume 1 molecule of NADH. And fructose 1,6-bisphosphate is dephosphorylated at position 1 to produce fructose 6-phosphate. Then fructose 6-phosphate is isomerized into glucose 6-phosphate. Finally, glucose 6-phosphate is dephosphorylated at position 6 to produce glucose. Enzymes of gluconeogenesis. The irreversible reactions in glycolysis are catalyzed by different enzymes that are unique to gluconeogenesis. All the reactions in glycolysis are reversible reactions, except 3 reactions which are known as irreversible reaction, catalyzed by hexokinase, which catalyzes the first step in glycolysis, and the reaction which catalyzed by phosphofructokinase 1, the key enzyme, and the reaction which catalyzed by pyruvate kinase, which catalyzes the last step. Only these 3 reactions need unique enzymes which is specific for gluconeogenesis. As we said, we have 4 precursors for gluconeogenesis. This figure shows the role of lactate in gluconeogenesis. Lactate either comes from RBCs because RBCs lack a mitochondria, and when it produces pyruvate by glycolysis, this pyruvate is converted into lactate. Also, muscle can provide lactate during exercise because glycolysis occurs anaerobically. This lactate is converted into pyruvate by lactate dehydrogenase. The pyruvate is enter the mitochondria, and instead of conversion into acetyl-CoA and enter-Krebs cycle, it converted into oxaloacetate by enzyme known as pyruvate carboxylase. But oxaloacetate cannot pass through the inner mitochondrial membrane. Is converted into malate, and malate go outside to the cytosol by malate shuttle. In the cytosol, malate is converted into oxaloacetate, and oxaloacetate is converted into phosphoenol pyruvate by phosphoenol pyruvate carboxykinase. The phosphoenol pyruvate go in the reverse direction of glycolysis until we reach fructose 1,6-bisphosphate. Then fructose 1,6-bisphosphate undergoes dephosphorylation at position 1 to produce fructose 6-phosphate, which is isomerized into glucose 6-phosphate. Then glucose 6-phosphate is converted into glucose. From phosphoenol pyruvate, the gluconeogenesis go in the reverse reaction of glycolysis until we reach fructose 1,6-bisphosphate. Another enzyme which is unique to gluconeogenesis catalyzes the conversion of fructose 1,6-bisphosphate into fructose 6-phosphate is known as fructose 1,6-bisphosphatase. This takes one phosphate group from position 1 of fructose to produce fructose 6-phosphate. Fructose 6-phosphate is isomerized into glucose 6-phosphate. Then glucose 6-phosphate undergoes dephosphorylation by a specific enzyme known as glucose 6-phosphatase, which is also unique to gluconeogenesis. This enzyme remove phosphate group from position 6 of glucose to produce glucose. By this step, gluconeogenesis is complete. The Curie cycle. It is the process by which lactate produced by muscle during strenuous anaerobic exercise is converted back to glucose through gluconeogenesis and then returned to the peripheral tissue. Muscle during exercise produce lactate under anaerobic condition by glycolysis. This lactate is taken through the blood to the liver. In the liver, lactate is converted back into pyruvate by lactate dehydrogenase enzyme, and the pyruvate is intergluconeogenesis. The pyruvate is converted into phosphoenol pyruvate. First, it converted into oxaloacetate, then into malate. The malate go outside to the cytosol and converted back to oxaloacetate, then into phosphoenol pyruvate, and go in the reverse direction of glycolysis until we reach fructose 1,6-bisphosphate. Then it converted back into fructose 6-phosphate by enzyme known as fructose 1,6-bisphosphatase. Then fructose 6-phosphate is converted, is isomerized into glucose 6-phosphate. Then glucose 6-phosphate is converted back to glucose by glucose 6-phosphatase. This glucose in the liver, which is synthesized from gluconeogenesis, is transported back to peripheral tissues. Conversion of glycerol to glucose. Glycerol is the second precursor for gluconeogenesis. Glycerol, which derives from the degradation of fat, actually triacylglycerol, is converted into dihydroxyacetone phosphate. First, the glycerol is phosphorylated at position 3 by glycerol kinase to produce glycerol 3-phosphate. Then glycerol 3-phosphate is converted into dihydroxyacetone phosphate by glycerol 3-phosphate dehydrogenase, which use NAD plus as coenzyme and produce NADH. The dihydroxyacetone is intermediate of glycolysis. This dihydroxyacetone phosphate combine with glyceraldehyde 3-phosphate to form fructose 1,6-bisphosphate. And then fructose 1,6-bisphosphate is converted into fructose 6-phosphate by enzyme known as fructose 1,6-bisphosphatase. بيحصل لها dephosphorylation. Then fructose 6-phosphate is isomerized into glucose 6-phosphate. Glucose 6-phosphate is converted into glucose by glucose 6-phosphatase. Conversion of amino acids into glucose. The third precursors for gluconeogenesis is known as glucogenic amino acid. The carbon skeleton of these amino acids provide various metabolic intermediates that can be used for glucose synthesis. For example, tryptophan can be converted into alanine, and alanine by transamination is converted into pyruvate. Pyruvate is converted into oxaloacetate by pyruvate carboxylase. Then oxaloacetate is converted into malate. Malate go outside to the cytosol by malate shuttle and converted back into oxaloacetate. Then oxaloacetate can convert it into phosphoenol pyruvate by phosphoenol pyruvate carboxykinase, and the phosphoenol pyruvate go in the reverse direction of glycolysis until we reach glucose. Another amino acids like glutamate can be converted into alpha-ketoglutarate by deamination, and the alpha-ketoglutarate is converted into succinyl-CoA, then into fumarate. Fumarate is converted into malate. Malate go outside to the cytosol by malate shuttle and converted back into oxaloacetate. Then oxaloacetate can converted into phosphoenol pyruvate and go in the reverse direction until we reach glucose. Glucose-alanine cycle. This is the relationship between liver and muscle. Alanine serves as a shuttle for pyruvate from the muscle to the liver. Pyruvate produced by glycolysis in muscle is transaminated to alanine. Alanine is transported to the liver where it is reconverted to pyruvate and used for gluconeogenesis. When gluconeogenesis produces glucose, this glucose is transported back to the muscle to be used as source of energy. Conversion of propionate to glucose. The fourth precursor for gluconeogenesis is known as propionate. The propionate comes from catabolism of odd number of fatty acid is first activated into propionyl CoA by enzyme known as acyl CoA synthetase, which use CoA-SH and also need ATP. Propionyl CoA is converted into dimethyl malonyl CoA by enzyme known as propionyl CoA carboxylase, which use ATP also as source of energy, and add carbon dioxide. It adds one carbon atom to carbon 2 of propionyl CoA. Then dimethyl malonyl CoA is converted to its isomer L-methyl malonyl CoA by enzyme known as methyl malonyl CoA racemase. This enzyme transfer the C double bond O of carbon 2 in dimethyl malonyl CoA from right to the left. Then the L-methyl malonyl CoA is converted into succinyl CoA by methyl malonyl CoA isomerase, and the succinyl CoA is intermediate of the Krebs cycle. It converted into succinate, then into fumarate, and the fumarate is converted into malate. Malate go outside to the cytosol by malate shuttle and converted into oxaloacetate. Then oxaloacetate is converted into phosphoenol pyruvate. The phosphoenol pyruvate go in the reverse direction of glycolysis until we reach fructose 1,6-bisphosphate, which is converted into fructose 6-phosphate by fructose 1,6-bisphosphatase. Then fructose 6-phosphate is isomerized into glucose 6-phosphate, and glucose 6-phosphate is converted into glucose. Condition associated with high gluconeogenesis rate. Condition that gives rise to high concentration of substrate of gluconeogenesis are high fat diet, because when we eat high fat in our diet, this provides glycerol and propionate, and those are precursor for gluconeogenesis. Also, the prolonged fasting and starvation, this stimulate the alpha cell of pancreas to secrete glucagon, which enhance the enzymes of gluconeogenesis. Prolonged strenuous exercise, this produce lactate, which is the substrate for gluconeogenesis. Glycolysis and gluconeogenesis goes in reverse direction. Glycolysis starts with glucose and end by pyruvate, and gluconeogenesis starts with pyruvate and end in glucose. The intermediate of glycolysis, fructose 6-phosphate, is converted into fructose 1,6-bisphosphate by phosphofructokinase 1. We have another enzyme known as phosphofructokinase 2. This converts fructose 6-phosphate into fructose 2,6-bisphosphate. It adds phosphate group in position 2 instead of position 1. This product, fructose 2,6-bisphosphate, very important in regulation of glycolysis and gluconeogenesis. It activates the phosphofructokinase 1, that means it activates glycolysis, and it inhibits the fructose 1,6-bisphosphatase, the enzyme of gluconeogenesis. That means it inhibits gluconeogenesis. References for this lecture include Harper's Illustrated Biochemistry, Stryer Biochemistry, Lippincott Illustrated Reviews Biochemistry, Usmali Kaplan. عندنا more references. عندنا لينينجر، ديفلين لو عايزين more details. النت كمان مفيد في بعض الأشياء. عندنا على قناة اليوتيوب في simplified videos ممكن تكون مفيدة. تم الكلام وربنا محمود، وله المحامد والعلا والجود، ثم الصلاة على النبي محمد، ما ناح قمري وأورق عود. This is all I can say in this lecture. I hope you understood. Thank you for listening and you have a great day.